Welcome, dear students. Today our topic of deliberation is Cladistics, Concepts and Terminology. The objectives of today's deliberation are to describe major terms and concepts used in cladistics, Plesiomorphic and apomorphic characters, homology and analogy, parallelism, convergence and reversal, monophily, paraphily and polyphily, cladogram. The second objective is to briefly describe the basic assumptions in cladistics. The last objective is to highlight the significance of cladistics. Cladistics is a quantitative method that attempts to recover evolutionary relationship based on observable characters and presents the resulting phylogeny in the form of tree-like diagram called cladogram. The basic idea behind cladistics is that members of a group share a common evolutionary history and are closely related, more so to the members of same group than to other organisms. These groups are recognized by sharing unique features which were not present in distant ancestors. Today, cladistics is a method of choice for classifying life because it recognizes and employs evolutionary theory. A central point to the determination of phylogenetic position of a particular group is the number of primitive, that is, plesiomorphic, or advanced, that is, apomorphic characters that the group contains. In cladistic analysis, species are grouped into natural or monophyletic groups based on sharing of syn-apomorphic homologs, while plesiomorphic homologs are rejected as valid evidence for relationship. Cladistic classifications are characterized by containing only strictly monophyletic, while specifically rejecting paraphyletic groups or polyphyletic groups. Now, let's have a brief introduction of cladistics. Cladistics or phylogenetic systematics is a branch of systematics concerned with inferring phylogeny, the evolutionary history or pattern of descent of a group of organisms. The term cladistics was coined by Kane and Harrison in 1960 and referred it to a relationship expressing recency of common ancestry. Sokal and Snead, 1963, described it as cladistic relationship refers to parts of the ancestral lineage and therefore describes the sequence of branching of ancestral lines. Ever since Darwin laid down the fundamental principles of evolutionary theory, one of the major goals of the biological sciences has been determination of life's history of descent. Cladistic methods are largely based on the principles of parsimony, according to which the most likely evolutionary route is the shortest hypothetical pathway of changes that explains the pattern under observation. Dear students, before analyzing the methodology of handling data for cladistic analysis, it's important to understand the main terms and concepts used in cladistics. So, let us discuss our first objective, that is, description of major terms and concepts used in cladistics. Let us first have a look at plesiomorphic and apomorphic characters. A central point to the determination of phylogenetic position of a particular group is the number of primitive, plesiomorphic or advanced apomorphic characters that the group contains. In the past, most conclusions about primitiveness were based on circular reasoning. These families are primitive because they possess primitive characters or character states, and primitive characters or character states are those which are possessed by these primitive families. Over the recent years, a better understanding of these concepts has become possible. It's generally accepted that evolution has proceeded at different rates in different groups of plants so that among present-day organisms, some are more advanced than others. The first step in the determination of relative advancement of characters is to ascertain which characters are plesiomorphic and which are apomorphic. Given insufficient fossil records of the earliest angiosperms, comparative morphology has been largely used to decide the relative advancement of characters. Many doctrines have been proposed, but unfortunately, most rely on circular reasoning. Some of the important doctrines are described below. The doctrine of conservative region holds that certain regions of a plant have become less susceptible to environmental influence than others and therefore exhibit primitive features. The doctrine of recapitulation holds that early phases in development are supposed to exhibit primitive features, that is, ontogeny repeats phylogeny. The doctrine of teratology was advocated by Sahani, who argued that when a normal equilibrium is upset, an adjustment is often effected by falling back upon the surer basis of past experience. Thus, teratology or abnormality is seen as a reminiscent of some remote ancestor. The doctrine of sequence advocates that if organisms are arranged in a series in such a way as to show the gradation of particular organ or structure, then the two ends of the series represent apomorphy and plesiomorphy. The doctrine of association advocates if one structure has evolved from other, then the primitive condition of the derived one will be similar to the general condition of the ancestral structure. Thus, if vessels have evolved from tracheids, then the vessels similar to tracheids, that is, vessels with longer elements, smaller diameter, greater angularity, thinner walls and oblique end walls represent a more primitive condition than vessels with broader, shorter, more circular elements with horizontal end walls. The doctrine of common ground plan advocates that the characters common to all members of a group must have been possessed by the original ancestor and must therefore be primitive. The doctrine of character correlation, the distribution of plesiomorphic character is displaced towards primitive members, which have higher proportion of plesiomorphic characters than the average for the group as a whole. The concept of apomorphic and plesiomorphic characters in understanding the phylogeny of angiosperms has been considerably advanced with the recent development of cladistic methods. Dear students, you might have understood what are plesiomorphic and apomorphic characters. Now let us try to understand the meaning of homology and analogy. When Charles Darwin first proposed his idea of natural selection, he used most evidence based on anatomical features of organisms. Two different ways of these similarities in anatomical structures can be classified as either analogous structures or homologous structures. Taxonomic groups or taxa are constructed based on overall resemblances. The resemblances due to homology are real, whereas those due to analogy are generally superficial. A real understanding of these terms is thus necessary in order to keep organisms with superficial resemblances in separate groups. These terms were first used and defined by Owen. Owen defined homology as the occurrence of same organs in different animals under every variety of form and function. He defined analogy as the occurrence of a part or an organ in one animal which has the same function as another part of organ in different animal. If applied to plants, the leaves of pitcher, Venus flytraps, poinsettia and cactus show different functions and shapes. Each of these leaves is a homologous structure derived from a common ancestral form. The tuber of potato and the tuber of sweet potato, on the other hand, are analogous. While potato is a modified stem meant for storage of food, sweet potato is a modified root. In the recent past, phylogenetic interpretation has been applied to these terms. Simpson defined homology as the resemblance due to inheritance from a common ancestry. Analogy similarly represents functional similarity and not due to inheritance from a common ancestry. Mayer similarly defined homology as the occurrence of similar features in two or more organisms which can be traced to the same feature in a common ancestor of these organisms. It is, as such, imperative that homology between two organisms can result only from their having evolved from a common ancestor, and ancestor must also contain the same features or features for which the two organisms are homologous. Wiley has provided a detailed interpretation of these terms. Homology may either be between two character states, or between two organisms for a particular character or character state. Two characters or character states are homologous if one is directly derived from the other. Such a series of characters is called an evolutionary transformation series, also called a morphocline or a phenocline. The original pre-existing character or character state is termed plesiomorphic and the derived one as apomorphic or evolutionary novelty. Three or more character states may be homologous if they belong to the same evolutionary transformation series. The term plesiomorphic and apomorphic are, however, relative. In an evolutionary transformation series representing the characters A, B and C as displayed in the figure, B is apomorphic in relation to A, but it is plesiomorphic in relation to C. Two or more organisms may be homologous for a particular character if their immediate common ancestral also had this character. Such a character is called shared homologue. If the character state is present in the immediate common ancestor but not in the earlier ancestor, that is, the character state is derived one, the situation is known as synapomorphy. If the character state is present in the immediate common ancestor as well as in the earlier ancestor, that is, an original character state, the situation is known as symplesiomorphy. The homology between different organisms is termed as special homology, as represented by different types of leaves in different species of plants. Different leaves in the same plant, such as foliage leaves, bracts, and floral leaves, would also be homologous, representing serial homology. The following criteria may be helpful in identifying homology in practice. Morphological similarity with respect to topographic position, geometric position, or position in relation to parts. Similar ontogeny. Continuation through intermediates, as for examples, vessels having evolved from tracheids. The primitive forms of vessels are more like tracheids, with elongated, narrower elements with oblique ends walls. When the same relatively similar character is found in large number of species, it is probably homologous in all the species. Sets of characters may similarly be homologous. If two organisms share the character of sufficient complexity and judged homologous, other characters shared by organisms are also likely to be homologous. Dear students, you are now familiar with the concepts of homology and analogy. Now let's have a look at parallelism, convergence, and reversal. Unlike homology, if the character shared by two organisms is not traced to a common ancestor, the similarity may be the result of homoplasy, sometimes considered as a synonym of analogy. It can result in three different ways. Number 1. Parallelism. Parallelism implies a similarity in biological makeup of ancestral forms. If the common ancestor of two organisms were not very ancient, and if the evolution in descendant lines followed more or less same course, then the term parallelism is used. The term is usually applied when the organisms have a common ancestor, but the character state was not present in their common ancestor. Simpson defined parallelism as the independent occurrence of similar changes in groups with a common ancestry as because they had a common ancestry. Number 2, convergence. Convergence implies increasing similarity between two distinct phyletic lines, either with regard to individual organ or to the whole organism. Similar feature in convergence arise separately in two or more genetically diverse and not closely related taxa or lineages. The similarities have arisen in spite of lack of affinity and have probably been derived from different systems of genes. Homoplastic traits are seen as convergent if they appear in distantly related groups or are realized by distant developmental mechanisms. Number 3 is reversal. Reversal is a loss of particular character, thus reverting to ancestral condition. The occurrence of reduced unisexual flowers without perianth or with reduced perianth in Ementiferae was once considered to be primitive situation, but the evidences from wood anatomy, floral anatomy and palynology have shown that apparent simplicity of these flowers is due to evolutionary reduction, that is, reversal. All the three situations, that is, parallelism, convergence and reversal, represent false sin apomorphy because the similar character state is derived, not traced to a common ancestor. Dear students, now let's understand the concepts of monophily, paraphily and polyphily. Cladistics construct cladograms, that is, branching diagrams, to graphically depict the groups of organisms that share derived characters. Key to cladistic analysis is identifying monophyletic groups, that is, groups comprising a given species, all of the species descendants and nothing else. In phylogenetics, a group of species is said to be paraphyletic if the group contains its most recent common ancestor, but does not contain all the descendants of that ancestor. A group comprising members from separate evolutionary lines is called polyphyletic. Evolutionary taxonomists consider polyphyletic groups to be errors in classification, often occurring because convergence or other homoplasy was misinterpreted as homology. Cladistics argue, therefore, that the prevailing classification system, lenient taxonomy, should be reformed to eliminate all non-clades. Further, cladists argue that paraphily is as harmful as polyphily. Therefore, cladistic taxonomy requires taxa to be clades, monophyletic groups. An excellent representation of monophily, paraphily and polyphily is presented by the cutting rules devised by Dahlgren and Rasmussen. The distinction is based on how the group is separated from a representative evolutionary tree. A monophyletic group is separated by a single cut below the group, that is, it represents one complete branch. A polyphyletic group is separated by one cut below the group and one or more cuts higher up, that is, it represents one piece of branch. A polyphyletic group, on the other hand, is separated by more than one cut below the group, that is, it represents more than one piece of a branch. Dear students, let's now try to understand the meaning of a cladogram. The end result of cladistic analysis is a tree-like relationship diagram called a cladogram. A cladogram represents an evolutionary diagram utilizing cladistic methodology, which attempts to find the shortest hypothetical pathway of changes within a group that explains the present phenetic pattern using the principle of parsimony. In a cladogram, all the organisms lie at the leaves, and each inner node is ideally binary. The two taxa on the either side of a ciplet are called as sister taxa or sister groups. Each subtree is called a clade and by definition is a natural group, all of whose species share a common ancestor. Each clade is set off by a series of characteristics that appear in its members but not in other forms from which it's diverged. These identifying characteristics of clades are its sin apomorphies. Each bifurcation of the cladogram represents a past speciation that resulted in two separate lineages. Dear students, with this we have reached our second objective, that is, basic assumptions in cladistics. There are three basic assumptions in cladistics. Assumption 1. Any group of organisms are related by descent from a common ancestor. This is a general assumption made for all evolutionary biology. It essentially means that life arose on Earth only once and therefore all the organisms are related in some way or other. Because of this, we can take any collection of organisms and determine a meaningful pattern of relationships provided have the right kind of information. Again, the assumption states that all the diversity of life on Earth has been produced through the reproduction of existing organisms. Assumption 2. There is a bifurcating pattern of cladogenesis. This assumption is perhaps the most controversial. That is, the new kind of organisms may arise when the existing species or populations divide into exactly two groups. There are many biologists who hold that multiple lineages can arise from a single originating population at the same time, or near enough in time to be indistinguishable from such an event. The other objection raised against this assumption is the possibility of interbreeding between distant groups. Assumption third, changes in the characteristics occur in lineages over time. This final assumption is the most important assumption in cladistics. It's only when characteristics change that we are able to recognize different lineages or groups. Dear students, now let us deliberate about our last objective, that is, significance of cladistics. Cladistics is now accepted as the best method available for phylogenetic analysis because it provides an explicit and testable hypothesis of organisms' relationships. Knowing the pattern of descent in the form of a cladogram can be viewed as an important end in itself. The branching pattern derived from cladistic analysis may be used to infer the collective evolutionary changes that have occurred in ancestral descendant populations through time. The cladograms can also be used to classify life in a way that directly reflects evolutionary history. Once a robust cladogram is derived, the pattern of relationship and evolutionary changes may be used for a variety of purposes like phylogenetic classification, studies on adaptation or speciation, biogeography, and molecular clock dating. The most striking uses of cladistics are: Number 1. Cladistics is used for creating systems of classification. Consider the bewildering variety of organisms that have ever lived on Earth, from jellyfish to bacteria. How is it possible that paleontologists, let alone other biologists, are able to communicate their ideas about such diverse topics as the history of life? Well, it's obvious that a system of classification is needed. That is, we need words like beetle or conifer so that we can talk about many organisms at one time. The second application of cladistics is that cladistics predicts the properties of organisms. As with any other system in science, a model is most useful when it not only describes what has been observed, but when it predicts that which has not yet been observed. Cladistics produces hypotheses about the relationship of organisms in a way that, unlike other systems, predicts properties of the organisms. This can be especially important in cases when particular genes or biological compounds are being sought. Such genes and compounds are being sought all the time by companies interested in improving crop yield and disease resistance and in search for medicines. Only a hypothesis based on evolutionary theory, such as cladistic hypothesis, can be used for these endeavors. The third application of cladistics is that cladistics help to elucidate mechanism of evolution. Unlike previously proposed systems of analyzing relationships, cladistics is explicitly evolutionary. Because of this, it's possible to examine the way in which characters change within the group over time, the direction in which characters change, and the relative frequency within which they change. It's also possible to compare the descendants of a single ancestor to look at the patterns of origin and extinction in these groups, or to look at a relative size and diversity of groups. Perhaps the most important feature of cladistics is its use in testing long-standing hypotheses about adaptations. For many years, since even before Darwin, it has been popular to tell stories about how certain traits of organisms came to be. With cladistics, it's possible to determine whether these stories have merit or whether they should be abandoned in favor of a competing hypothesis. Dear students, this brings us to the end of lecture on cladistics, concepts and terminology. Hope you enjoyed the lecture.